before also, what does it mean? So gene therapy means when we are doing some therapeutic procedures on the gene, when we are and what by which means we can do it, we can use advanced biotechnological tools and techniques to correct the gene's function. That is called as gene therapy. Means if we are using any of the gene manipulations to correct any of the gene genetic defect, if to correct any of the genetic abnormality, that procedure is called as gene therapy. And this gene therapy can only be done by biotechnological tools. So here when we are talking about the applications of biotechnology, then we are talking about this gene therapy under healthcare system. So after birth, the correction therapy of the hereditary disease can also be possible now. So we can correct any of the gene defect after birth too, but it is very much it will, if we can correct this defect before birth, then that would be so much beneficial. Later in this topic, we will see how it is so much essential to correct the gene defect in embryonic period or during embryonic stage. So here after birth, the correction therapy of the hereditary disease can also be possible now. This therapy includes correction method to correct the gene defect into their cells. So in the cells, if any of the gene defect is there, if any of the hereditary disease is there, the hereditary, what is the meaning of hereditary disease? If any of the gene defect is passing from one generation to another, then it is called a hereditary disease. And this disease is only because of the abnormal gene. Okay, so here by this newly inherited gene, by this newly inserted gene, okay, what happens for the gene therapy? What we are doing, we are inserting a new gene. Means to correct the abnormal gene, we are inserting our gene of interest. Suppose a defective gene is there in a person, then what we are doing, we are inserting a construct. We are inserting our gene of interest. If a gene A is defective in a person and it causes some abnormal function in the body, then what we are doing, we will take the gene A from another human being, the normal gene sequence we will take as a gene of interest, and what we will do, we will try to put that gene construct in that particular human being. Then that particular inserted gene of interest, gene A, will start producing the normal protein, and that normal protein will correct the defect. So by this newly inserted gene, by the biotechnological tools and techniques, what it will do, it takes over the function of the defective gene. So whatever defective gene's function was there, if the gene is defective, it means the gene is not able to produce the normal protein. Gene is not able to produce the actual protein. And if the protein is not actual, if the protein is having some defective sequences of the amino acid, then it will not be fully functional. Even though sometimes it will not be functional too, okay, then it will not be able to perform its function. So what happens when we are inserting a gene, then that particular gene, that inserted gene or gene of interest, will take over the abnormal gene's function. And after that, what will happen? It will make the abnormal cell to the normal one, because it will start expressing their own protein. Now, one of the examples of the gene therapy we will take here as a severe combined immunodeficiency syndrome, or SCID. In short, we are using the term SCID. So the full form of this SCID is severe combined immunodeficiency. Okay, so by the name we can understand what would be the result of this disease. Okay, so what happens in this disease? Persons lose the immunity power, and if the person is losing immunity power, it means the person's body's defense mechanism will be affected. Okay, so here this severe combined immunodeficiency is caused by a defective gene, ADA. Okay, ADA is a gene. If this ADA gene is becoming defective, if the DNA sequence of this gene ADA is not proper, is not normal, then it will not be able to produce its own protein. and if this protein will not be produced, then it will cause a disease. This disease is called severe combined immunodeficiency. Okay, so what happens due to defect in ADA gene? This skid happens, and what is the full form of this ADA? Adenosine deaminase. So adenosine deaminase is a gene which is producing normally a protein which is actually involved in the immune system, and if this protein will not be expressed properly, if this protein is not fully functional, then person will be affected by this disease skid. So we will see the history of this disease, how scientists tried to correct this disease in the affected person. So in 1990, first gene therapy was given in a 4-year-old girl with ADA deficiency, means adenosine deaminase deficiency, means that girl, that 4-year girl was affected by skid by the abnormal gene, this ADA. Okay, so the deficiency of this protein is caused by the deficiency of ADA gene. So if the ADA gene was defective, it means this girl was not able to produce the adenosine deaminase protein, and ultimately this disease was happened. So in 1990, first gene therapy was came in existence by correction of this gene. So you will have to remember the year 1990 was the year in which the first time the gene therapy was taken into the consideration, and the disease first gene therapy was done for skid disease, and the targeted gene was adenosine deaminase, ADA. Okay, and when we talk about its function, then we could see here which is crucial for the immune system to function. I told you that this gene is involved in the immune system. Okay, and if gene is not normal, it means it is hampering the function of our defense mechanism or immune system. So it is hampering the our immune power. Okay, so what they did, what scientists did, bone marrow transplantation and injection of ADA protein could not be completely curative. So first what they did, first what doctors or scientists tried, what they did, they took ADA protein from the outside, filled in the injection and injected in the bone marrow. Okay, so what happened when the protein was injected, then up to a certain time it was curative, but it was not completely curative, because after some certain time point the whole protein was consumed for its function. Then again that girl, since it was it was affected by a genetic disease, it means again that girl will be in deficiency of this particular protein. So all the time you cannot inject the protein all the time in the bone marrow. It is so much difficult procedure, and it is so much painful procedure to inject anything in the bone marrow, and it is not also allowed to disturb the bone marrow, because sometimes it can cause cancer also. So what happens, first time what scientists did, they took the ADA protein and injected directly in the bone marrow. Then after some time that protein was utilized, and the immune system was becoming normal. But after some time, when the whole ADA protein which was injected in the bone marrow consumed completely, after that again the same condition occurred. Skid means the immunodeficiency was again, you could see in that girl. So the gene therapy procedure was undertaken. So after injecting protein, we could understand that this is not the ultimate solution of this genetic disease. So what we did, I'll show you. So what scientists did, they isolated the lymphocyte cells. Okay, they isolated the lymphocyte cells of the patient. After that, they cultured. Means I told you the meaning of culture, cell culture. They grown the lymphocyte cells in the flask. After that, they proliferated. They increased the number of the lymphocyte cells of that patient girl. And after that, what they did, they took a recombinant gene. You just see here they have taken this recombinant DNA, and in this recombinant DNA they have put the gene of interest. And what was the gene of interest? The defective gene. So cDNA, complementary DNA of ADA gene, they inserted in a expression vector. And what they did, this recombinant DNA, they inserted in the lymphocyte cells. So what they did first, they took the lymphocyte cells from the patient girl. After that, what they did, they proliferated, or we can say they cultured the cells to increase their number. After increasing the number, what they did, they took the recombinant DNA in which our gene of interest was there. And what was the gene of interest in this case? The cDNA, complementary DNA of the ADA gene, and they inserted in the expression vector and formed the recombinant DNA. And this recombinant DNA they inserted in the lymphocyte. And after that, after inserting this the recombinant DNA into the lymphocyte cells, what was going on? The lymphocyte started expression of the protein which we inserted deliberately. So these lymphocyte cells will now start producing the product of this ADA gene. So these lymphocyte cells which are having this recombinant DNA will start producing the ADA gene. Okay, so now here this lymphocyte introduced in the then what happened? The expression vector introduced in the lymphocyte. This lymphocyte started the production of adenosine deaminase protein. And after production of this, when they checked the expression, after that they took the lymphocyte, and the lymphocyte was injected in the girl's body. So here previously the product was injected, but after that, after seeing that all the proteins has been consumed, after some time, after some days, the whole protein will be consumed, then they inserted the recombinant DNA in the cell, in the particular cell, means the lymphocyte cell. And these lymphocyte cells in cultured medium, they took and injected in their bone marrow. So what happened after that? The lymphocyte will start producing the protein. Okay, now this lymphocyte in the girl's body will start producing the adenosine deaminase protein. Then up to a certain limit, this disease will be cured. But there is a limitation. Okay, since lymphocytes are not immortal, so regular infusion of such genetically engineered lymphocyte is needed. So again, one condition came, as after that each of the cell is having its own life cycle. After some time, after its lifespan, it will die. Like that, the lymphocyte we had injected in the bone marrow of that girl will die after some time. So whatever lymphocyte, genetically engineered lymphocyte, whatever we have injected in the girl's body, after some time they will start dying. And after their complete death, what will happen? When each of the injected lymphocyte will die, then again this disease will occur. Okay, so this is not a permanent solution. But as compared to the injection of protein, means as compared to protein injection, this would be so much beneficial, as the injection of this genetically engineered lymphocyte will work for a long time as compared to direct injection of a particular protein. But it is not a complete solution. So in conclusion, what we are saying, at early embryonic stage, introduction of such DNA in their stem cell, bone marrow, could be a permanent solution, could be a permanent cure of this disease. So what happens in early embryonic period? If we can get the information that this baby is having this disease or may have this disease, if the person is going through the diagnosis of genetic diseases, and we can find that this embryo is having this disease, then in embryonic stage, if we are doing some gene manipulations, then we can permanently cure that particular disease by this genetic engineering. But after death, it is so much difficult to cure completely. All the time you will have to provide the genetically engineered lymphocyte. So by this example, you will have to understand certain things that the genetic here in case of gene therapy, it was started in 1990. What gene was targeted? Adenosine deaminase gene. What was the disease which we tried to cure by the biotechnological application? SCID, severe immunodeficiency. And and you will have to remember that this point because it is very much essential to know that if we are doing some gene manipulations in embryonic state, then it will be permanent solution. That's it. Now we will see another application of the biotechnology in healthcare system is molecular diagnosis. What is the meaning of diagnosis? If we can detect any of the disease, then that is called as diagnosis. So when we are diagnosing the disease based on the biomolecules, then it is called as molecular diagnosis. And biotechnological tools and techniques are one of them. So by using biotechnological tools and techniques, we can go through the diagnostic procedure using the molecules as well as the gene. Means at gene level, at protein level, and biomolecule level, whatever diagnostic procedure we are using will come under the biotechnology. Okay, you must have heard about the RT-PCR. In case of this COVID, we are going through the RT-PCR. RT-PCR is one of the application of the biotechnology. You must have heard about the ELISA. So different nowadays we are using different tools and techniques in case of healthcare system and especially in molecular diagnostic procedures. Okay, so we will see here one by one. With the improvement in our knowledge about the reason of pathophysiology of the different disease, it is impossible to diagnose every disease by simple histological or histopathological analysis or serum analysis or urine analysis or blood analysis. We know that some biochemical tests are there using which we are detecting and analyzing different disease, but it is impossible to detect all the diseases and the reason of the diseases by using these simple techniques. Histopathological samples means what we are doing. What in this section, what we are doing, we are just putting the tissues or the slide or the transfer section of the cell, and by observing the abnormality within the cell, we are confirming that this person is having this disease. By serum analysis, we are detecting some of the proteins, some of the biomolecules by using different reagents. We are detecting the amount of abnormal secretion of anything, like in case of diabetes, we are detecting the level of glucose in the blood, level of glucose in urine. So by biochemical test, we cannot diagnose all the diseases. Okay, so there are certain limits with histological analysis, serum analysis, urine analysis. So recombinant DNA technology, polymerase chain reaction, that is in short is called as PCR, enzyme linked immunosorbent assay. Okay, this is in short called as an ELISA. You will have to remember the full form of all these shortcuts. Okay, PCR means polymerase chain reaction, ELISA means enzyme linked immunosorbent assay. So all these diagnostic procedures come under the biotechnology. Okay, has become the useful tool for disease diagnosis. These techniques are more accurate and reliable too, because we are using the molecular level diagnostic procedures. That's why these are so much reliable as compared to simple biochemical test. By using simple biochemical test, what we are doing, we are using some reagents, we are putting different reagents in the sample, and based on the color, based on their concentration, based on different things, we analyze the disease, but those are not reliable. But in when we are using these techniques, PCR, ELISA, or certain biotechnological tools and techniques, then those are much more reliable for the disease diagnosis. In case of, you can compare the both of the procedures. You just see some, in case of this coronavirus, we are uh, we are realizing more on RT-PCR, real-time PCR, as compared to rest of the diagnostic procedures. So what we are doing, based on biochemical test, although we can detect certain diseases, but when we are going through the biotechnological tools and techniques in case of diagnostic procedure, then those are so much reliable. Even though when you are not having any symptom, if a few virus or few bacteria has infected you and symptom has not been produced also, then also you can use these biotechnological tools to detect the less number of viruses and bacteria too. Means not other techniques can do it. Okay, so by using PCR you can detect a single virus in your body also. Okay, that's why we are going through the RT-PCR test, because suppose I am not having any symptom of the corona, but if I am going through the RT-PCR or qPCR test, then if any of the single virus is there in my body, then it will be detected. But if I am going through another procedure, that antibody test is there, so that antibody test, sometimes it gives false positive and false negative result also. But in case of RT-PCR, it is so much reliable. That's why a lot of agencies need RT-PCR tests result. Okay, now we'll see what are those techniques and what we can do with this. Okay, so first biotechnological diagnostic tool is PCR, polymerase chain reaction. You will have to remember the full form. Okay, so in case of PCR, what we are doing, we are using primer. Okay, we are using primer for a specific gene. Primer means what? You have already studied the primase gene. Primase gene is making the primer. Okay, that primer initiates the DNA replication. So what happens? We are using the primer for a specific gene, and that primer amplifies the particular gene. Suppose if we want to detect the coronavirus, then we will use certain stretch of the DNA of that particular virus, means coronavirus. We will use the primers of coronavirus that generally is not present in case of human being. Means that virus is having its own gene. So if we are making primers based on the RNA sequence of the coronavirus, then it will bind to the DNA of DNA or RNA of coronavirus. As you know that due to reverse transcription, RNA of virus will be converted into DNA, and that DNA will be inserted in our genome for their replication. So when we are going to detect any of the gene, any of the pathogen, then we are trying to make the primer based on the pathogen. So here, using primers of a specific gene of a pathogen, so that gene should not present in the human being. So that some of the bacterial genes are present only in bacteria. Some of the genes are present only in virus. So we are targeting those genes. Okay, so based on those genes, means based on the specific genes of the pathogen, we are making primers. And what we are doing after that, we are going through a procedure in which we are replicating that particular segment, that is called as PCR. So here, using primers of a specific gene of a pathogen could not only detect only with small amount of population, but also their amount also. Means here, by using PCR techniques, two benefits are there. First thing, we can detect small number of viruses. If very few viruses have infected us, then we can detect it. First thing, first benefit. The second benefit, we can detect its amount also. Means how many viruses had infected my body, we can detect it. So two things we can do with the PCR. First thing, we can detect the small number of viruses. That detection cannot be done by any other procedure other than the post PCR. The second benefit of the PCR is to detect the amount of infectious material in our body. Means suppose I'm infected with hundreds of virus, and another person is infected with thousands of virus, and another person is infected with the millions of virus. Then we can detect their amount also easily by using this PCR technique. We can detect the number of these viruses. We can get the information as how much virus, how much of bacteria, how much pathogen is present in the body. Even it is present or not, it can give the result. Okay, this is a very sensitive method. Even can detect the presence of virus, bacteria, etc. when symptom is not visible. I told you, if bacteria or virus is present in my body, and if it is not creating any symptom also, suppose that sometime when the virus infects our body, then it takes some time for 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 showing any symptom. But before any symptom, we can detect the presence and absence of virus. That's why we are going through RT-PCR test for coronavirus, and where it has been employed. So we could see here that PCR technique we are now using in case of detection of AIDS, in case of detection of this cancer and corona also. So all these are important diseases in which we are using the PCR technique. Okay, so you will have to remember certain things in PCR. We need to have the primers to amplify the particular gene of the pathogen. Using that particular primer, we can amplify them, and we can get the information about their infection, either infection is there or not. First question. The second question, we can get the answer. The second question, how much infectious material is there in your body, means how many viruses have infected your body. We can detect it. Now it is so much sensitive technique, and we can detect virus, bacteria, and any of the pathogen who is having the nucleic acid. Okay, so PCR is a nucleic acid based technique, means we are amplifying the gene. Okay, and the 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 diseases in which we are using the PCR technique: AIDS, cancer, corona, and several diseases are there. Now, the second technique is ELISA. I told you the full form of ELISA. Yeah, and here we will talk about this ELISA procedure. So here I told you the PCR is genetic based techniques, means it is DNA or RNA or nucleic acid based technique. But when we are talking about this ELISA, then we are talking about the antibody based technique. So you will have to remember that PCR is a nucleic acid based technique, and here it is antibody based techniques. Means here we are detecting the antigen based on antibodies, and here we are detecting the presence of gene based on different primers. Okay, so here full form of the ELISA is enzyme linked immunosorbent assay. You will have to remember the full form. Okay, now antibody produced against a specific pathogen in non-human species such as mice, rabbit, monkey. What we are doing, suppose if we want to make the antibody for the coronavirus, so what we will do, we will take the coronavirus, will inject in either mice or rabbit or monkey. Okay, so when we are injecting any of the pathogen in any organism, like these are the experimental animals, है ना? Mice, rabbit, monkey, guinea pig, different species of the organisms are there, which would be used in our ELISA procedure. So what we are doing, we are directly injecting the pathogen in their body: mice, rabbit, monkey, guinea pig, etc. And when we are injecting any of the pathogen in their body, then what will happen? After some time, these organisms defense system will start producing antibody against that particular virus. If any of the foreign material enters in your body, then our body's defense mechanism system, or we can say the lymphocytes of our WBC, will start producing antibodies against that. Okay, so if we suppose we have taken a rabbit, and in that rabbit we have injected the coronavirus, then the rabbit's body will start producing antibodies against that coronavirus. Okay. After some time, what we will do? We will take the blood from this rabbit, infected rabbit, and with the blood we can collect the antibody which has been formed against the coronavirus. And you know that antibody works in a specific manner. Means if we are injecting coronavirus, then antibodies will be produced against coronavirus only. If we are injecting the any of the bacteria in the rabbit, then rabbit's body will start producing antibody against that particular bacteria. If we are injecting the poliovirus in rabbit, then rabbit's body will start producing antibody for against the polio virus. So these antibodies are so much specific, means these antibodies encounter only the particular pathogen. Okay, so what we did, we took rabbit, we injected coronavirus, then the rabbit's body started producing antibodies, and these antibodies we have collected by blood collection. And after that we took the antibody, and on that antibody what we did, we tag the signal molecule. Signal molecule means we can tag a fluorescent molecule. Okay, so what we will do, we will attach a signal molecule. It means whenever we will get this antibody, then this signal molecule will produce some fluorescence. By the presence of this fluorescence we can get information that this antibody is there in this tube. Okay, so you can easily detect the pathogen and patient sample with antigen antibody binding. Okay, so signal molecule gives the information about binding and non-binding condition. So what will happen, so we have collected the antibody, we have from the rabbit, which has been formed against the antigen, or we can say antigen is that virus. Antigen is any of the foreign material, either it is virus, bacteria, or any of the protein. So rabbit's body has formed a lot of antibodies. On that antibody we have attached a single signal molecule. Now what we will do, suppose we have taken the blood or the sample from the infected person. If suppose a patient is coming and giving the sample for the detection of coronavirus, so what we will do, we will take the blood from that person, and what we will do, on we will immobilize the blood here on the surface. After that what we will do, since we have produced antibodies against that particular antigen, means against that coronavirus, then we will allow to attach that antibody. So if that antibody finds antigen, means virus, then it will get attached, it will get adhered. Got it? And suppose this patient is devoid of that infection. If patient is not infected with the coronavirus, then the blood of that person will not have the corona antigen. Then the antibody which has been produced against that particular virus will not be able to bind it. Then after some time what will happen, since this antibody has not been attached on the blood of that negative sample, then it will be removed off. So after some time what will happen, in positive, it means in infected person, we will get the signal, we will get the fluorescence here. And in case of negative sample, means the person who is not infected with virus will give will not give any kind of signal. I'll show you the whole thing by this. I'll show you the whole thing, and on this platform you just see, suppose a patient is coming with the coronavirus, then what we will do, we will collect the blood and put the blood in, just a minute, just a minute. Okay, so if a patient is coming with a viral infection, then what we will do, we will have this. Okay, just a minute. So what we will do, we will take the blood sample. In this blood sample, viruses are there. So suppose these are the viruses. So these are the viruses present in the blood, and since we have collected the antibodies which have been produced against that virus, then what will happen? These antibodies will bind over the surface of these antigens. Means what will happen? Antigen antibody binding will happen. And since we have attached a signal molecule over this antibody, so due to this signal molecule, we can detect its presence. So in the sample, if antibodies are still there after washing, then it means the person's blood is infected with the coronavirus. Got it? And in case of a person who is not having the coronavirus, then suppose we have taken the blood. So this one is the blood, and in this blood no coronavirus is there. These are the coronaviruses, so coronavirus is not there. Then what we will do, we will take the antibodies, we have taken the antibodies which was collected previously from the infected rabbit, so we will allow to adhere. Okay, and on this antibody signal molecules are there, so we will allow to adhere. So since no virus is there in this blood, what will happen? Antigen antibody binding will not happen. So here binding will not happen. So what will happen? Whatever antibodies we have allowed to adhere with the virus will not adhere because of the absence of viruses. So after washing, what will happen? All these antibodies will be washed off. So all these antibodies will come out of this wall, with this well. So when we will try to detect it, then in this well, nothing will happen. Means no signal will be there because no attachment is there. No attachment is there just because of the absence of antigen and antibody binding. Why antigen antibody binding has not happened? Just because of absence of coronavirus. But in this case, you just see in positive sample here. In case of positive sample, since virus was there, antibodies have been produced against that virus, so they will obviously attach with that, and due to this attachment, antibodies will be still there after washing also. And since they are having the signal molecule, so by the fluorescence, when we will put under some machines, under UV radiation, under the detection machine, then machine will detect the fluorescence. Due to this fluorescence, we can easily detect. Okay, either we can get the signal molecule, can be of anything. Either they will be fluorescent, either they will produce some color with a substrate. If we are putting some chemical which will become the substrate, then after putting the reagent, it will give a color. So in this well, there will be some color, and here, since no antibody is there, antibodies has not been attached with this blood, after washing it will come out, and when we are putting the reagent, then it will not give the color. So it will be colored and it will not be colored. So due to presence and absence of this color, or due to presence of signal molecule and absence of signal molecule, due to fluorescence and due to non-fluorescence of the wells, we can easily detect a particular disease. So in this diagnostic procedure, we have talked about the PCR as well as the ELISA. Okay, so here